Hi everyone. Coming to you recorded from my lab. I gave you this PowerPoint Google Slides not too long ago, so I just wanted to give you a quick rundown of what I'm expecting from you so that you have a really good handle on cells. For right now, we don't know what learning is going to look like going forward. As soon as I have more information, I will pass it on to you. In the next week, please look for a Zoom meeting invitation so we can at least get together and visit a little bit and answer any questions you have, and I can help you find the next thing to move on to. So, on to the slideshow. I hope that you've done this. A big part of what we're starting to learn and understand right now about learning from far away is that you're doing it for yourself. You're doing it for your own understanding and your own growth. You're not doing it for me. It was never the case that you were doing the work for us. So keep that in mind as you go forward. Ask yourself, do I understand this? Can I answer questions? When I write questions out, am I answering them completely? Am I writing sentences that make sense in standard English? I think a lot of times we slap down answers, try to use the right keywords, but don't actually read it. So I'm really going to ask you to be introspective, look into yourself and think about, how could I do this better? How could I make this more? How could I understand this? Any concepts or topics or vocabulary you really don't understand, just post in the comments and I'll get back to you as quickly as I can. I'm online most of the day, not necessarily sitting here at a desk waiting for you, but checking in consistently to see if anybody needs anything. So for each slide, I hope that you've copied the diagram onto a piece of paper. Drawing is going to help you remember what things look like. Cells are hard because a lot of things are circles, so you can kind of give it a little bit of character, and we'll see that in a minute when we get to the gifts from the Amoeba Sisters. Label only the structures in the box. I think each cell coming up has like 4 or 5 structures, so just label those and just describe the job that you listed. And then this part is important. Read your description out loud. Hear yourself saying the words. If you have another person, if you have a grown-up or your dog even, or your cat or this bunny, you can go ahead and just say them to that organism, and hopefully as they're coming out of your mouth, you hear it. You either hear that it makes sense or you hear that it doesn't. All right, so here's our animal cell. The words are ribosome, nucleus, lysosome, mitochondria. So we hope that you know that ribosome makes proteins. It's going to take a genetic message from the DNA. We're going to copy that into RNA. We're going to send it to the ribosome, and that's how that protein is going to get made. The only proteins we can make are proteins we have instructions for. So that includes all your pretty features, your skin color and your hair color and your eyelash style. Do they come straight out or curl up, or what do they do? But also all of the enzymes that you need to digest your food, all of the instructions you need to build new blood vessels or new skin cells when you get a cut. So we go back to the nucleus. The nucleus has all those instructions in DNA. That's really all it does. Remember, it's just a pocket to keep your DNA. Lysosomes, these are our cleanup crew. So these are going to be packed with enzymes. So if we go back real quick to the nucleus and the ribosome, in the nucleus are the instructions for how to make digestive cleanup enzymes. Those instructions have to get passed somehow using mRNA as an in-between molecule to the ribosome. The ribosome makes the enzyme. It's going to get some help. We're going to come back to that in a minute. And then those get packaged in lysosomes, so now we've got a cleanup crew. But again, the instructions all have to be there. And lastly, our powerhouse of the cell, mitochondria. Mitochondrion is one, mitochondria is plural. And remember, those do cell respiration and they make ATP. When we look at our plant cell, I just included a couple of the really specialized organelles that are unique to plant cells. Our cell wall is going to help the plant cell provide structure, especially when water is low. It also, we learned, protects the cell against diseases. So there are viruses, just like coronavirus, that can attack plants, and a plant cell wall helps keep that virus away from the cell membrane. Our cell membrane, remember, it's going to control what goes in and out. It's really going to let things like oxygen and carbon dioxide go right across. Water, so-so, but big molecules and things like viruses it should keep out. Viruses have their own tricky ways, though. Chloroplast, these are always going to appear green. Unfortunately, when I print for you, I can't print in black and white, but when I do make you a quiz or an activity, I can give you a colored picture. Chloroplasts will always be green. They're collecting the sunlight energy and they're doing photosynthesis. They're making sugar. With that sugar, we're going to send it to the mitochondria and have it broken down to make ATP. It always has to come down to ATP. Lastly is our large central vacuole. This should be full of water most of the time, and it's really pressing everything up against the edges of the cell to keep it nice and stiff. The trick is, though, if in low water conditions, this starts to shrink and the cell can get really floppy, and that's where the cell wall will come in. All right, so I made this slide. This is a little crazy. I hope it works. So here's a big animal cell. So first we're going to pass through the cell membrane to get into the cell. That was Riley. And once we get in, look what I did. I made it. If you look at this in the not presentation style, it looks like a mess. Once we get in, we get into the cytoplasm, which is mostly water. It's kind of got the texture of really loose, unfinished jello. It's kind of gross, but it's crowded. The cell is full of organelles. But in order to get a job done, all the cell parts have to work together. So let's get to work. We're going to go right to the nucleus. Again, that's where the instructions are. So we're going to make an mRNA copy of the DNA, and we're going to send that out to make our protein. The way we're going to do that is we're going to send that mRNA copy to the ribosomes. They're going to take the gene instructions, and they're going to literally chain up amino acids together to make a protein. However, our protein needs to be folded, so we're going to send that to the rough ER. You can see on the rough ER that there are some of these same purple dots of ribosomes. It doesn't matter where the ribosomes are, they will make the protein, and then it'll go into the ER to get folded and shipped around. If it's a cell that's going to ooze something out of it, like your intestines might do, we're going to send that protein we just made to the Golgi body. The Golgi is going to package it all up in a way that's going to allow the cell to ship it outside, so out into this kind of tan space here. So here's our vacuole, and the Golgi is going to package up our protein in the vacuole. That vacuole is going to meet up with the cell membrane, and our protein will end up on the outside of the cell. And that's it. So those are the major pieces of what the cell is doing all the time. The only bit that changes a little is whether or not the protein is shipped outside the cell or is kept inside the cell. Like I said a minute ago, sometimes we package up like cleanup enzymes and we put them here in this lysosome, and then that's just going to float around freely and clean up any messes that happen to get made. Ta-da! So I animated each of those arrows independently. So if you were to look at that slide in the not presentation view, it is a mess. Don't worry about that. Just go to the presentation view and keep clicking. Once again, here's some things that you can do. There's no grades right now happening, but we really want you to learn for learning's sake. Find the joy in learning. Start to discover what's interesting about all of this. These are two very simple readings, the soft schools. This is a Quizlet on organelle cell functions. And then Cells Alive has a great model that you can kind of click through and play with and see some organelles. All right, so here are the questions. These are MCAS questions. So we're just trying to look at some of these and think about what they might be asking. Antibiotics are medicines used to treat bacterial infections. Some antibiotics work by interfering with the bacteria's ribosomes. Other antibiotics work by interfering with the bacteria's plasma membrane. Describe the function of ribosomes and explain why interfering with them would kill the bacteria. So from before, we remember that ribosomes make proteins. If the bacteria can't make proteins, they can't do anything. They can't reproduce. They can't break down sugar for food. They can't make ATP. They really can't. They're kind of locked. They're stuck. So if we attack the factories, the whole cell will shut down. The other kind of antibiotics interrupt the plasma membrane. So remember, that's your only barrier between the outside world and the inside of the cell. So if the antibiotics disrupt the cell's membrane, the cell's just going to kind of like explode or mush or fall apart into pieces, and it won't be a cell anymore. So those are two ways that antibiotics can help you if you have a bacterial infection. Remember, viruses aren't bacteria, and so you can't do these things because they don't have these things. This was the second half of the question. Medicines called antifungals are used to treat infections caused by fungi. One way antifungals work is by targeting cell parts that are present in fungal cells but not in human cells. So here we have to tack back onto the taxonomy that we just studied, looking at the difference between animals, plants, fungi, and protists. Other than a ribosome or a plasma membrane that human and fungal cells have in common, well, they have a nucleus, they have mitochondria, they have ERs, they have Golgis, they have lysosomes, they have, it says ribosomes, they have all of the same eukaryotic cell parts in common. Describe what would happen to a human cell if it were affected by an antifungal. So first and foremost, antifungals actually affect the fungus cell wall. Fungi have a special cell wall that's very different from animals. That's why we can use that kind of drug, because it interferes with a type of cell part that fungus have that animals simply don't. However, the interior of the cell, the inside, it's all the same everywhere. So, for example, if a drug targeted the mitochondria, there's no difference between a fungus mitochondria and a human mitochondria. So if we had a drug to try to get rid of a fungus like athlete's foot that targeted mitochondria, your cells would lose energy too, and that would be really bad. So think about what cell part you chose and think about what job it does in both types of cells and why we really can't interrupt the functioning of that. So just these are super fun pictures of each organelle that kind of give you their gifts, right? They animate so they show you. We got Power Hands here. He's making the energy, right? He's crackling with energy like a superhero. We've got our lovely green chloroplast who is making all this glucose with the sun. We've got our lysosome. This is a bacteria minding his own business, but the lysosome isn't having any of that. We've got our nucleus. With great power comes great responsibility. It's got all the instructions. Here's our ribosome. It's reading the mRNA, and he's being a factory, putting out the amino acid, plus he's got a sweet mustache. Here's our little ribosome factories stuck on our rough ER, and the cell is assembling all those proteins and making them packaged and folded. Next, we've got our Golgi. He's packaging the proteins, sending out these little packages like a little Amazon shipment center. Our vacuole is storing stuff like water, and then cytoplasm, we're just filling up the cells so you can see all the stuff inside. I know you love it. Play this game again if you really want to master cells. I'm going to come back to you with cell energy next time. So please stay strong. This is me being sworn in as Constable of Swampscott. And I'll see you soon. We'll have a Zoom meeting just to hang out. Talk to you soon, everyone. Bye. Stay safe, stay healthy, stay home.